do vaccines. So let's talk about some kinds of new vaccines. And this is going to be a presentation that I'm using that is from Ian Jones at the University of Reading. He was kind of my mentor when I was there. He's actually worked in vaccine development, done some very interesting things with that. And he taught a good course on this and said it was okay if I used it. So we're going to do it. Okay. So the idea here is that. So we're going to talk about vaccine types and principles of protection. We'll look at what happens following infection and immunization. We'll take a look at some new vaccines, and we're going to focus on particularly rotavirus and dengue virus vaccines. We can mention the human papillomavirus vaccine. We're not really going to talk about the pneumococcal vaccine because that's going to be bacterial, but these three are going to be viruses. And actually, we've covered the, well, no, we'll cover those first three in a little bit of detail, but spend most of it on the first two. Okay, so let's go down. All right. Okay, so there are actually different kinds of vaccines in the world. The kind that you're going to be most familiar with are going to be prophylactic vaccines. These are going to induce neutralizing antibody, which are antibodies that are able to bind to the virus and shut it down, stop it from infecting. And so they're going to prevent the pathogen from actually getting into the body in the first place. So when you're taking the flu vaccine or something like that, you are aiming for a prophylactic vaccine. That's the main idea. But there are also a couple other vaccines, therapeutic vaccines. So for example, if you're bit by a rabid animal, they can actually vaccinate you with the prophylactic vaccine. And because it takes so long for the rabies virus to actually work its way through the body to the point where it's a problem, you can actually develop protective immunity before the virus gets to your brain. And yeah, it's fascinating and really wonderful that we're able to do this. Okay, so vaccines have to change from year to year because viruses change as well. And so shown here is the influenza hemagglutinin protein, just as a model. And you can see on the outside, we've sort of highlighted with these little ball and stick models, each of the amino acids that actually changes from one year to the next. And you notice what they have in common. They're all located in these little loops at the edge, and they're on the outside of the hemagglutinin. It's not changing the things at the center of the hemagglutinin. This is where it would bind to its receptor. It's not really changing that stuff at the bottom. This is the fusion motor with heptide repeat 1 and 2 down in here. No, it's changing the parts that are actually there in order to change. Yeah, the C-terminal region, this sort of red part down here, is the fusion motor. And so it has a job to do, and it can't just randomly mutate. But all this stuff up here is basically decoration. It's there so it can mutate, and it's just this little pocket right in the center that's actually going to bind the sialic acid. And that part doesn't really mutate either. So there are parts of the virus that do a job and they don't change, and there are parts of the virus that are just there in between and they do change. However, if you look at total amino acid substitutions that occur over all the different strains that we know, you can see that they are distributed. There are still some parts, like this fusion motor back in here, that really do not pick up many mutations at all. But eventually, yeah, you can change almost anything on the outside of the virus and still come away with an infectious virus. Those are the places where mutations are tolerated, and these in the center are the places where mutations are not tolerated. That's the idea. Okay, so vaccine strategy in general boils down to these three. There are live vaccines, which we call attenuated, which means these are weakened strains. So it's a virus that still is able to work, but it has some kind of genetic problem that means that it doesn't grow particularly well or it doesn't cause disease. And so this is kind of the earliest version of a virus vaccine, a modified vaccine that was made, and it's still very successful. There is, however, a risk because since it's a live virus and it's only a few nucleotides, mutations different from the virus that actually causes disease, there are times when this virus can change. Viruses mutate all the time, and it can actually go back to the virulent form. So another way that's a little safer but a little less effective is to use chemically treated or killed virus. The benefit with a live virus is that you get all of the genes being expressed in the normal way, and you can set off every single part of the immune system with a live virus. With a killed virus, you get parts of an immune response, but not as much. And so sometimes people, it's totally safe to use, but sometimes people won't make enough of a response that it actually works. So if you would have got the flu vaccine, you would have probably got a killed, chemically treated version of that. And then we also have this modern way of doing it, which is with engineered split or recombinant vaccines. And so some of the flu vaccines are split. They will take this live virus and just break it up into little pieces, or they will just make one part of the virus that the immune system is able to recognize, but they can't hurt you. Or in some cases, they'll just put in DNA to make one part of the virus that will then theoretically go on to educate your immune system so that it can resist the full virus when that does come. So the current vaccines for things like the oral polio vaccine are actually based on forced evolution. So what you do is you take the virus that's fully able to cause disease and you start putting it into cells where it doesn't normally grow. And then you take it out of those cells and put it back onto those same kind of cells where it doesn't ordinarily grow. And then you keep doing this, and every once in a while you're going to switch it up. You're going to change from one kind of cells to a different kind of cells, and you're going to force the virus to pick up mutations because the virus is going to do its best to be the optimum at whatever it's trying to do. And once it puts in a mutation, it has a very hard time actually getting that mutation out. And so if you keep running it through all these weird little tests, like a crazy little Olympics, at the end you can get out a virus that actually has a whole bunch of mutations. And you can test it out, and quite often the virus will be attenuated. It won't be able to cause the same disease. Because first it's got good at growing in this kind of cells, and then this kind of cells, then this kind, in a little flask without any immune system around. And that's actually very different to what it would have to do inside of an actual person. And you don't want this to be just like one nucleotide away, because of course it can make that quasi-species and this sort of cloud of viruses around it, and it can get right back to full virulence. So that wouldn't be very good. But yeah, the most successful vaccines like oral polio vaccine, the BCG vaccine, so this is for tuberculosis, there we go, and the MMR vaccine, measles, mumps, and rubella, are all based on selection of attenuated strains, so actual live viruses, but weakened viruses that are able to grow, but we've trained them to grow somewhere else, not in a person, and so they're very bad at growing in people. Okay, the advantages of attenuated vaccines as opposed to these other ones, they activate every part of the immune system. Totally right. So you can get IgG in the bloodstream, that's what humoral means, and you can get IgA, which is an immunoglobulin. These are both antibodies at the mucosal surfaces, like in the intestine or in the lungs, for example. And so that's a good way to get protection against catching the virus again. And so, yeah, you tend to have durable immunity. It will last for years, maybe even for life. And you tend to get a little bit of cross-reactivity. So you can make a response against one virus if it's a live virus, and that can sometimes help you fight off other related viruses. It's low cost. All you have to do is grow the virus. That's very easy. And yeah, you tend to get quick immunity in these vaccines. And so, right, this can be used to drive other vaccines, or rather other viruses to extinction. This is the strategy that drove smallpox to extinction. This is the strategy that's currently driving poliovirus to extinction. Okay, but there are other kinds as well. So let's talk about subunit or split vaccines. So they're only going to be made from part of the pathogen. Usually, with a virus, it's going to be one or two of the proteins, often just one protein. Some will be isolated directly from the virus. They'll just grow up live virus, break it into pieces, and take out the pieces that they want and throw away the rest. They're going to take out the parts that give you a good immune response and throw away all the parts that could be dangerous or probably wouldn't give you much of an immune response. For example, if you've been vaccinated for hepatitis B, that is going to be just the surface antigen, just the surface protein of hepatitis B expressed in yeast. And this is what they will use to inoculate you and vaccinate you. These split or subunit vaccines have no genetic material whatsoever inside them because they're only just a part of the virus. They're not the entire intact thing. And so they're not going to replicate themselves, and they're not going to be able to spread or mutate. And so they're actually very safe to use. And so in order to do this, you have to get some knowledge of which parts of the virus are going to be most helpful. And this takes a bit of trial and error. The downside, though, is that they're often rather poorly immunogenic in that they don't really make good immune responses sometimes. So the immune system responds more if it is more sick, if it's dealing with something more like a real virus. And these vaccines may or may not give enough protection, but they will definitely be safe. So there'd be some situations where you would want to err on that side of definite safety. And there'd be other situations where you would definitely want to have a something like a weakened version of the virus, if such a thing exists and is stable. Okay. What are the disadvantages of live attenuated vaccines? Yeah, we've covered these. So they can mutate and they can turn back into the virulent form. And sometimes it's pretty easy to do. And then once they turn back into the virulent form, or even if they don't, they can spread from the person you vaccinate to people who have not wanted to be vaccinated. This could be an advantage in communities where not everybody gets vaccinated, but you don't want to be sort of stealth vaccinating people, I guess. I don't know. That seems a little bit weird. There are there are other problems, but those are the big problems. Now, what's the disadvantage of the inactivated vaccines? Yeah, many of the people who are vaccinated will not get immunity. With some of these vaccines, they only work in about 40 or 50% of the people who are actually vaccinated will be protected to some extent. You also tend to need boosters for these, which means you have to get vaccinated more than once because your body just doesn't get enough of a chance to make an immune response with one go. Sometimes you have to have 2 or 3 or 4 of these courses. And if you have a pet, you'll know that they sometimes have to go back for multiple vaccines. It's the same idea, booster shots. Inactivated vaccines also tend to not produce local immunity. So you tend to not get immunity at the site where you put the vaccine in. So, for example, you wouldn't get those antibodies on the inside of the lungs or on the inside of the intestine. And because this is a thing that has to be manufactured at a biotech firm, you're probably going to end up with a higher cost, and therefore there will be some people that can't afford to get this thing. And if they mess up with the inactivation, then you can end up with virulent virus there. It doesn't happen much anymore, but it has happened in the past with polio vaccine batches. This is a long time ago, like 1950s, if I remember right. Okay, in the next section, we're going to look at how a vaccine actually works, and then we're going to pause this section. Okay. So, first of all, there are these things called B cells. And B cells' goal is to get good enough at recognizing virus that they can actually end up making antibodies. And so the B cell has B cell receptors, and these are like trial antibodies. These are all the things it's trying out. These are all its, weird high school poetry or whatever. And it's making mutations. It's combining new things together to make its B cell receptor. And if it happens to get a hit, if it happens to be able to stick to the virus, then the B cell is going to get a reward. We're going to, if we get more than one of these B cell receptors very close to each other, then we're going to activate a thing. It's going to be a tyrosine kinase based activation. That's going to tell the B cell it's time to divide. You've done it. Yeah. Success. Your poetry was a hit. Yeah. But then, of course, there are other cells that are going to recognize antigens in different ways and present them. And so in the body, there are going to be these dendritic cells. Remember, they're out in the periphery, and they look kind of like an amoeba. The dendritic cell's job is to find anything that doesn't look right, any little bits of virus or just any sign of trouble, and then bring it and show it to a T cell. So remember, dendritic cells are like the mall cops, just patrolling the periphery out in the middle of nowhere. But when they find something, they don't deal with it themselves for the most part. They're going to bring the evidence to their supervisor, which is going to be one of these things like a T cell, and they're going to show it. And they can show it directly to a killer T cell, and the killer T cell can go in and kill whatever it is that made it. Or they can show it to a helper T cell that can go and help educate a B cell, or go help support those other T cells and work in that way. And so that's going to be the other main presentation way. Now, a third thing that actually makes these vaccines work, pardon me, is going to be these proteins called toll-like receptors. And so toll-like receptors are going to be abbreviated as TLR. And there are several different toll-like receptors that can interact with parts of viruses. So these are proteins that are going to be, some are on the surface of the cell, some are actually inside the cell. So if a cell has eaten something that seems to be, a little bit wrong, like a virus particle, these would be ways to detect that. And they're going to be set off by different things. So TLR 7 and 8 are going to be set off by single-stranded RNA. Because if a cell is eating RNA, that's weird. That kind of sounds like you just ate a virus. CpG containing DNA, which is kind of strange. But there are a lot of viruses and bacteria that would have something like this. And TLR9 is going to be set off by that. Some viral proteins will set off TLR4, and double-stranded RNA, which is another thing you'll find in some viruses, is going to set off TLR3. And so these are going to be, if these TLRs signal, these are going to be located, I believe, on the dendritic cell. Then the dendritic cell will move with more urgency, and it'll be more likely to activate a strong, they'll activate like a strong presentation, basically. They'll do a good job of presenting this, and we'll get lots of activated T cells and B cells. Now, there's a lot more detail than we're going through here, but that's more like for immunology class. And so we're going to save that mostly for later. We just want an overview. TLRs exist, and this is how they work. And so these are some of those TLRs. There are all these proteins. Some are on the surface that bind different things from bacteria or viruses. Some are on the inside of different cells. And so if they notice a virus that happens to have burst on the inside of a little lysosome, then they can detect some of the components inside and start the cell reacting. Yeah. And then once the TLRs are activated, they actually have this long signaling cascade. And we're not going to learn it. I'm just going to show it to you right here. So we're going to set it off. The key component here is going to be a thing called MyD88. And MyD88 is in most of the TLR signaling pathways, but not all. Some like TLR3 are going to signal through TRIF. But anyway, in the end, we're going to make things like IRF3 and NF-kappa B. And these are things that you've heard of before. They're going to go into the nucleus, and they're going to start that interferon A gene program. They're going to get the cell ready to make interferon and receive interferon signals, and they're going to get the cell ready to shut down viruses. They're going to make all those tetherins and other proteins like that. Okay, there are actually a couple other sensors inside the cytoplasm, and you already know some of these, right? So inside a dendritic cell, you're going to have RIG-I and MDA5, which are going to be looking for, we think, poorly capped RNA. So those are definitely there. And then remember that cells are going to have MHC class 1 and MHC class 2. These are, MHC class 1 is going to be equivalent to HLA A, B, and C. And MHC class 2 is going to be equivalent to the HLA-D, and there's 3 versions of that. These are going to be for talking among cells of the immune system, and MHC class 1 is going to be for talking like regular cells communicating with the immune system. And so these are all ways of presenting the peptides. And so if you make an antigen, and the antigen goes in there, so you're vaccinating, for example, then there's going to be a signal from the components that's going to go through something like RIG-I, which is going to have an effect. We're going to make some cytokines, for example, and we're going to have actual responses to the antigen itself. We're going to chop that up into little pieces, load it on the outside for MHC class 1 or 2, and then we will present that and get a really nice immune response. This is the idea. Okay, let's move past that. And then, yeah, just if you wanted to see how this happens. So infection itself, like live infection, is going to further stimulate the immune response. So if you have all these things, if you're making a RIG-I response at the same time as you're presenting peptides, overall this is going to lead to more cytokines being made, which will call in more T cells and say, hey, look at what I've got. And you're going to also make these things called co-stimulatory molecules, which are like a little reward that you can give for T cells that are able to dock with these little peptides and read them out and basically find something that matches there. Okay, and so we'll look at these in the next section, and we'll pause right here.